Right now, inside your body, your immune system is likely being tricked by sugar.
Yeah, literally tricked.
I mean, we usually think of cancer's ability to evade our immune system as this like hyper-complex genetic cloaking device, And sure, it is, but at the physical level, tumors are actively coating themselves in a dense forest of sugar molecules to basically just put your immune cells to sleep.
It's a brilliant and honestly devastating survival mechanism. I mean, we've developed these revolutionary immune checkpoint therapies over the last decade, right?
The big game changers.
Exactly. These are drugs designed to wake the immune system up so it can attack the tumor. But for a frustratingly large number of patients, the drugs just stop working. The tumor adapts.
Which brings us to the core mystery we're diving into today. Like, why do some tumors just shrug off our absolute best immunotherapies? And to figure that out for you, we are looking at some groundbreaking new research that actually maps out the genetic factories building these sugar disguises.
It's fascinating stuff.
It really is.
Yeah.
But to understand the disguise, we have to talk about the glycome. So for everyone listening, imagine the surface of a cancer cell. It is not it's not a smooth, round balloon.
No, not at all.
It looks more like a dense, overgrown jungle. And instead of vines and leaves, that jungle is made of complex carbohydrate structures.
Right. And that is your glycome. And hidden within that sugar jungle, cancer cells will overproduce one very specific type of sugar called sialic acid.
Okay.
We call this process hypersialylation. The sialic acid basically acts like a biological do not disturb sign on a hotel door.
And who exactly is walking the halls reading those signs?
That would be your immune system's security guards, specifically a family of receptors called Siglecs.
Siglecs.
Right, which stands for sialic acid-binding immunoglobulin-type lectins. It's a mouthful.
Yeah, let's just stick with Siglecs.
Good call. So these are inhibitory receptors patrolling your body. When an immune cell bumps into a tumor cell and its Siglec receptors bind to that dense canopy of sialic acid, it triggers a signal.
A stand-down signal.
Exactly. It tells the immune cell to stand down and just hold its fire.
My first instinct here is pretty simple. Like, if we know the tumor is using these specific sialic acid leaves to hide, why don't we just find the biological factory making the leaves, take an axe to it, and shut it down? Just, cut off the sugar.
I wish. Biology rarely makes it that easy, unfortunately. The genetic networks that shape these sugar structures operate with immense redundancy.
Redundancy meaning backups.
Massive backups. There isn't just one enzyme building the disguise. There are entire families of enzymes with overlapping jobs. So if you develop a drug that blocks one enzyme from adding a sugar molecule, another enzyme from a completely different family just steps up to the assembly line and covers the shift.
It's like biological whack-a-mole. You hit one, and three more just pop right up.
That is the perfect way to describe it. And this redundancy completely broke our older methods for studying this system.
How so?
Well, for a long time, researchers used something called CRISPR interference, or CRISPR. You essentially go into the cell, break a specific gene, and see if the tumor loses its sugar disguise. It's a loss-of-function test.
But wait, if there's a backup enzyme waiting in the wings, breaking the first gene won't actually change anything, right? The disguise just stays intact.
Exactly. You look at your data and falsely conclude that the gene you broke wasn't important, when in reality, it was just masked by the backup system.
Oh, wow. That's a huge blind spot.
It is, which is exactly why the researchers in today's study had to flip the script entirely. Instead of breaking genes to see what happens, they used a massive gain-of-function strategy called CRISPR.
The A stands for activation, right?
You got it.
Okay, let's unpack this. Because the paper mentions they used let me read this, DCAS9-based transcriptional activation system coupled with a SUNTAG VP64 scaffold.
Yeah, that's a lot of jargon.
I am going to need you to translate that for me.
Yeah.
Like, what does a SUNTAG actually look like physically inside the cell?
Sure, let's break down the machinery. So the DCAS9 part stands for dead Cas9. Normal CRISPR uses Cas9 like molecular scissors to cut DNA, right?
Right.
Well, here they basically broke the scissors. The DCAS9 acts like a homing beacon. It finds the specific gene, binds to it, but doesn't cut.
So it just parks there on the DNA.
Exactly. And attached to that parked homing beacon is the SUNTAG scaffold. Think of it like a long molecular extension cord with multiple sockets on it.
Okay, extension cord, I like that.
And into those sockets, they plug in these activator proteins called VP64. The end result is a massive amplification signal. You are forcing the cell to read that specific gene and crank out the corresponding protein at absolute maximum volume.
So if the old loss-of-function method was like firing a worker on a factory floor to see what breaks, this new CRISPR activation method is like giving every single worker a massive megaphone and like a dozen energy drinks to see what they are uniquely capable of shouting over all the background noise.
Yes, that's a perfect analogy, and the scale of this experiment was staggering.
Tell me about the scale.
They took K562 cells, which are a type of human leukemia cell, and they infected them with 104,000 different guide RNAs.
104,000.
Yeah. These guides directed the megaphones to systematically force the upregulation of 20,800 individual genes, one by one.
Okay, so you have this massive pool of thousands of cells, each with a different gene screaming at the top of its lungs. How do you actually figure out which of those shouting genes is successfully building the sugar disguise?
You introduce the security guards.
Ah, the Siglecs.
Right. The researchers engineered soluble Fc-fused versions of Siglec 7, Siglec 9, Siglec 10, and Siglec 15.
You know, they basically created free-floating versions of the immune receptors, not attached to a cell.
Yes, exactly. And they attached a fluorescent tag to them. So they watched these glowing security guards over the massive pool of leukemia cells.
I see where this is going.
The cells that had upregulated a gene responsible for building a great sugar disguise would bind really tightly to the glowing receptors. Then they fed the whole mixture through a machine called a flow cytometer.
Which literally uses lasers to look at cells one by one. Right.
One by one, in a single file fluid stream, the laser hits the cell, and if it glows brightly, the machine sorts it into a high-binding tube. If it doesn't glow, it goes into a low-binding tube.
And then comes the math.
Always the math.
They sequence the RNA of the highly glowing cells and use a statistical algorithm called CASTLE, that's C-A-S-T-L-E, to sift through the noise. It calculates exactly which megaphone genes significantly enhance the tumor's ability to grab onto the Siglec receptors.
That's the entire workflow, beautifully summarized.
Okay, so a quick bullet summary for you listening at home. Cancer hides using the sugar forest. Because of redundancy, we can't just chop genes out to see how the forest grows. So instead, researchers use CRISPR activation to give 20,800 genes a molecular megaphone, then washed glowing immune receptors over them to physically sort out which genes were building the strongest disguises. Think about the sheer scale of that for a second. Like, if you could give any biological process a megaphone to understand it better, would you look at how cells grow, how they communicate, or how they hide? Just something to think about.
It really reframes how we tackle biological mysteries. When you force a system into overdrive like this, you discover things that normally just hide in the background noise.
Right.
And what they heard from all those megaphone genes fundamentally shifted our understanding of how cancer builds these structures. There is no master switch for hypersialylation.
Right. It's not a single driver event at all. Multiple genetic roots converge. And that brings us to one of the most fascinating mechanisms in the paper, the galactose bottleneck. Let's focus on the immune receptor Siglec 7 for a minute.
Okay, so Siglec 7 is highly expressed on natural killer cells. Those are your body's first responders against tumors.
This front line?
Exactly. And to put those natural killer cells to sleep, the tumor builds a very specific sugar structure called a sialylated core-1 glycan. Now, these structures are built on top of carrier proteins on the cell surface, right? The paper mentions proteins like CD43 or mucins like MUC22 acting as the foundation.
They are the foundation. And building the sugar structure on top of them requires modifying a foundational sugar ring called a galactose residue. But here's the critical part. There's a fierce physical competition happening at the exact 3OH position on that galactose ring. So it's a biochemical tug of war.
A massive one.
You know, I picture this part like a really busy train switchyard. You have two rival factions of enzymes fighting over the tracks. On one side, you have the GLC and ACT branching enzymes, families like GCNT and B3GNT. What do?
Well, they want to add complex branches to the sugar structure, but when they branch it, they change the physical shape in a way that actually prevents the sialic acid from being added properly.
So they effectively ruin the disguise.
Right. They switch the tracks away from immune evasion. If you use the megaphone on the gene GCNT1, you actually reduce the tumor's ability to bind to Siglec 7.
But then you have the rival faction in the switchyard, the sialytransferases, specifically the ST3GL family.
Yeah, ST3GL1 and ST3GAL2. These enzymes want to cap the galactose ring directly with sialic acid. They are switching the tracks straight toward immune evasion.
And the algorithm picked that up.
Big time. The CASTLE algorithm showed they were incredibly potent enhancers of Siglec 7 binding.
And what's wild is what didn't show up. You'd think the genes responsible for manufacturing the raw material, like the actual sialic acid itself, would be huge hits.
You seem so, yeah.
But genes involved in CMP sialic acid biosynthesis weren't prominent at all. It basically proves the raw material isn't the limiting factor. The tumor has plenty of sugar. The entire system bottlenecks at that specific train switch yard on the galactose ring.
That is a crucial insight for drug development. But the screen revealed something even more surprising. Amidst all these expected capping enzymes, a completely unexpected gene emerged at the very top of the list for Siglec 7.
Here's where it gets really interesting, because that gene is GAL3ST4. And this blew my mind, because GAL3ST4 is not a sial transferase. It doesn't add a sialic acid leaf.
No, it doesn't.
It's a sulfotransferase.
Correct. Instead of adding a sugar, it attaches a sulfate group to that exact same contested spot on the galactose ring.
I need to pause here because this feels counterintuitive. If Siglec 7 is a receptor specifically designed to look for sialic acid, why on earth does adding a sulfate group suddenly make the tumor bind so much stronger? Like, how does that physically work?
It comes down to three-dimensional molecular geometry.
Okay.
A sulfate group carries a really strong negative charge. When GAL3ST4 attaches that sulfate to the core 1 structure, it alters the electrical landscape and the physical shape of the entire sugar molecule.
So it bends it into a new shape.
Exactly. It creates a highly specific configuration, a 3-sulfo-6-sialy core 1 structure. And that unique shape happens to be a mathematically perfect physical fit for the binding pocket of the Siglec 7 receptor.
It's like cutting a new groove into a key so it turns the lock flawlessly.
That is exactly it. And the researchers didn't just leave this as, a statistical anomaly in the data. They went into LN229 glioma cells, which are aggressive brain cancer cells, and they physically removed GAL3SD4.
And what happened?
The tumor's disguise basically collapsed. Binding to the Siglec 7 security guards dropped by roughly 80%.
80%, just from that one gene.
Yep. And when they engineered the cells to express GAL3ST4 again, the strong binding immediately returned.
Okay, let's lock in this picture for a second. The tumor's Siglec 7 disguise relies on a structural bottleneck. It's a literal fight between branching enzymes and capping enzymes at a train switch yard. And the surprise heavyweight champion in this fight is a sulfotransferase, GAL3ST4, which physically reshapes the sugar to perfectly fit the immune receptor's lock, especially in glioma cells.
That covers it.
So a question for you listening. Knowing that tumors use highly specific tissue-restricted tools like GAL3ST4 to build their disguises, how might this change the way we design targeted therapies for specific organs like the brain? We aren't just looking for chemicals to kill cancer cells anymore. We're looking for molecular wrenches to jam into very specific train switch yards.
It's a whole new paradigm.
But as soon as we map out one receptor, the cancer throws another curveball. We have to talk about Siglec 9 and Siglec 10 because the rules just completely change here.
They absolutely do. What's fascinating here is Siglec 7 is rigid and bottlenecked. Siglec 9 exhibits extreme structural plasticity.
Meaning it's flexible.
Very. It doesn't just bind to those core 1 glycans we just discussed. It will happily bind to N-glycans as well, which are completely different sugar structures attached to different amino acids on the carrier proteins.
The redundancy here is terrifying. When the researchers gave the megaphones to the ST3GAL family, almost every single one of those capping enzymes enhanced Siglec 9 binding.
Yeah, there was only one exception noted in the text, ST3GAL5. But otherwise, it's a massive overlapping network utilizing carrier proteins like CD44 and CSPG4 to basically present the disguise.
Which means if a pharmaceutical company spends $1 billion developing a drug to block just one ST3GAL enzyme, hoping to unmask the tumor to Siglec 9, it's just doomed to fail. 3 other enzymes will just step up and cover the shift.
Exactly. It's a hydra. But while Siglec 9 showed us frightening redundancy, the data on Siglec 10 actually dismantled a widely held scientific dogma.
Okay, I am so glad you brought this up, because I have a major point of confusion here based on recent literature.
Oh.
Yeah, I feel like for the last few years, every paper on Siglec 10 points to one specific carrier protein called CD24. The narrative has always been that CD24 is the ultimate don't eat me signal, like the master lock and key ligand for Siglec 10. Is that suddenly wrong?
It's a really great example of why we must continuously test our assumptions. The field heavily focused on CD24 because it is highly expressed in several solid tumors and it clearly interacts with Siglec 10. But this CRISPR screen was unbiased. It looked at the entire genetic landscape all at once.
And what did it see?
Well, in the K562 leukemia cells and the THP1 monocytes they tested, they blasted the megaphone on the CD24 gene. They overexpressed it massively, and the result,
Nothing.
nothing. It was completely dispensable for Siglec-10 binding in these cells.
Wow. So the old prime suspect is essentially cleared of the charges, at least in these specific blood cancer lines.
Yes, the data forces us to adopt a much broader model. Siglec-10 doesn't strictly rely on CD24. It actually recognizes a highly diverse, complex repertoire of alpha-2,3 and alpha-2,6 linked N-glycans across many different karyoproteins.
So the disguise is a mosaic, not a single mask.
Exactly, a mosaic.
It's fascinating how fast science corrects itself. To quickly summarize this shift, Siglec-9 utilizes a massive redundant network of ST3-GAL enzymes, except ST3-GAL5, on carriers like CD44 and CSPG4. Meanwhile, Siglec-10 reads a massive mosaic of diverse glycans, proving that the old suspect CD24 is actually dispensable in certain cell lines. So here's a question for you. When a widely accepted scientific model, like the CD24 and Siglec-10 connection, gets challenged by new data, how long do you think it takes for medical textbooks to actually rewrite the rules? It really makes you wonder what other single-target therapies and clinical trials right now are fundamentally misunderstanding the complexity of the tumor.
It's a humbling thought for anyone in the field.
Which brings us to the ultimate question.
Yeah.
So, what does this all mean? We now have this incredible spreadsheet of Castle-E scores. We have a map of genetic switches, redundant pathways, shape-shifting sugars. How do we get this out of a lab dish and into a patient's treatment plan?
If we connect this to the bigger picture, the immediate application is using this data set as a framework for predictive biomarkers.
So testing the patient before treatment.
Exactly. Before we give a patient an expensive Siglec-directed immunotherapy, we shouldn't just guess if it will work. Doctors could sequence their specific tumor and look at the expression of these exact networks.
You'd look for the factories. Like, if the tumor is highly expressing GCNT1, that's the enzyme that branches the tracks away from evasion, you might predict a really good response. But if it's overexpressing our surprise guest, GAL3ST4, you know the tumor is heavily disguised.
Precisely. And speaking of GAL3ST4, because it emerged as such a potent driver, it becomes a massive target for future drug development. Its baseline expression is naturally restricted in healthy human tissue, but it is highly enriched in specific cancers like glioma.
And the paper noted it upregulates ligands for both Siglec-7 and Siglec-9. It's a multitasking enzyme.
It is.
If you can block GAL3ST4, you might be able to strip away two different security disguises at the exact same time, elegantly bypassing a lot of that frustrating redundancy we talked about.
It is a highly compelling target. However, as with all groundbreaking science, we have to look really clearly at the limitations of the method. The authors themselves are very rigorous about this.
Right, because CRISPR is ultimately an artificial overexpression system. Giving a gene a megaphone doesn't perfectly replicate the natural environment of a cell.
Right. The technical term is stoichiometry. That's the exact natural ratio and balance of enzymes interacting in a physiological environment.
It's kind of like baking a cake. CRISPR might be dumping like 10 cups of sugar into the mixing bowl when the cell normally only uses the teaspoon.
Good analogy.
It shows you exactly what the sugar can do, and it highlights hidden players, but it's an engineered extreme. Also, as a gain-of-function screen, it struggles to tell us anything new about genes that are already screaming at the top of their lungs at baseline. You can't make the loudest worker in the factory that much louder.
Exactly, you hit a ceiling. The other major hurdle for clinical translation is the jump from in vitro to in vivo. Moving from single human cell lines in a dish, like the K562s, to living, breathing systems is messy. We rely heavily on mouse models in drug development, but human and mouse Siglec biology is incompletely conserved.
Meaning they just don't match up perfectly. A drug that brilliantly blocks a sugar factor in a mouse might fail in a human because our immune security guards have evolved slightly different locking mechanisms over millions of years.
Exactly. It's a classic translational bottleneck.
So if a drug cures a cancer mechanism perfectly in a mouse, but the biology isn't conserved in humans, how would you approach redesigning the experiment? It's the kind of puzzle researchers face every day.
It's the hardest part of the job, honestly.
Well, let's bring all these threads together. What we've learned today fundamentally changes the narrative of cancer immunotherapy. Cancer glycome remodeling is not a singular event that you can just find and switch off. It's a dynamic, highly redundant, network-based process. It's a constant physical tug-of-war between enzymes like sulfotransferases and sialytransferases, fighting for control over microscopic molecular real estate to build a dense forest of sugar that puts our immune system to sleep.
We are no longer looking for a single locked door. Thanks to technologies like CRISPR, we are finally mapping the entire wiring schematic of the building, and that map is going to guide the next generation of precision oncology.
And that leads me to a final provocative thought for you to carry with you. We now have this incredible genetic map of the factories building these sugar disguises, but all of this was mapped on isolated cells floating in a lab dish. What happens when you put those cells back into the chaotic reality of a human body?
That is the frontier of this research right there. Because the text mentions the tumor microenvironment. How does the physical three-dimensional structure of an intact tumor change the way these enzymes compete with one another in a real patient's body?
Yeah, think about the physical stiffness of tumor tissue, the chaotic blood flow, the severe starvation of oxygen inside a solid mass. How do those extreme, real-world physical stressors alter that delicate biochemical tug-of-war we talked about? Because a factory operates very differently when the building is under extreme pressure or practically on fire.
That's the next great mystery to solve.
The diagnostic landscape of cancer might be murky, but we are finally learning how to read the waters. Thank you for joining us on this deep dive. Keep questioning the complex networks operating just under the surface of the biological world, and we will see you next time.